Hi, y'all. Welcome to our first module in our workshop, Mushroom 101, Cultivation and Wild Foraging. This is our first of 5 modules in the series, and I'm your host, Amanda Lee. I'm so excited to have you here. And we're going to dive into my presentation, which is more structured like a lecture. And then towards the end, I will just be sharing from the heart, channeling my message to you all. So if you are a note taker, feel free to grab a pad and a pen, and we'll get into it here. So this is Module 1, Mushroom Basics, the Mycelial Mindset. Here's our lesson overview. So we're gonna get into the mycelial mindset, what mushrooms are, fungi, not plants, mycelium, the underground intelligence, ecological roles, decomposers, symbionts, healers, mushroom life cycle, safety and ethics, gear that is needed for cultivation, agar and inoculation, and then spore prints, spore swabs, cultures, and liquid cultures. So first of all, the mycelial mindset. Cultivating fungi begins with respect, not so much technique. Technique does come in, but we really want to cultivate a respect for the fungi kingdom. Fungi belong to an ancient kingdom with their own rules of life, what I like to call the mycelial mindset. So fungi and mushrooms, they're patient, they're interconnected, they're adaptive, and they're quietly intelligent. Fungi are not plants. Mushrooms belong to the kingdom of fungi, which actually was only discovered by biologists about 50 years ago. Before, people thought they were plants, but the more we've studied them, the more we've come to know that they do not photosynthesize. They digest their environment externally. Fungi break down organic matter using enzymes. Biologically, fungi are closer to animals than plants. They breathe oxygen, they exhale carbon dioxide, they transform matter instead of producing it. The hidden organism, mycelium. So you can see this picture here, this beautiful thread-like rope. That's what mycelium looks like. And you think of mushrooms, you think of the fruiting body, but the main organism is what's underground, and that's the mycelium, which is a vast network of thread-like cells which spreads through soil, wood, and organic debris. It functions as nature's recycler, communicator, and architect. Mycelium senses and responds to its environment. It navigates obstacles and finds nutrients. It shares resources with plants and other organisms. It forms living networks beneath ecosystems and encourages a shift from isolated thinking to network thinking. The ecological roles of fungi. So they are known as decomposers. They break down dead plants and trees and organic waste and then return nutrients to the soil. They are essential for ecosystem renewal. They are known as symbionts. They form mycorrhizal partnerships with plants. They exchange minerals and water for sugars. They support forest and agricultural systems. And they're healers. They produce compounds that protect ecosystems, and they also provide medicine used by humans for centuries. So here's a photo of the mushroom life cycle. You can see it goes from fruiting body to releasing the spores, growing into hyphae, and then the hyphae meet and combine to create mycelium. And then primordia is like a stronger version of the mycorrhizae that becomes the fruit body selection, and then the mushroom grows forth from this underground magical process. And so, just to go through this one more time, the spores disperse into the environment, they germinate into hyphae, hyphae fuse to form mycelium, mycelium expands through its substrate, environmental signals trigger fruiting, mushrooms release spores, and the cycle continues. And we're going to really be getting into the nooks and crannies of this process as we move through in our Q&A sessions. Here's another diagram of the life cycle of a mushroom. So you can just kind of see the process here. Inoculation to germination to expansion to primordia, formation, fruit bodies spring forth from that growth. And we are going to learn about cultivation, but cultivation as collaboration. So the mushroom is an expression of a hidden network, and growth responds to environmental conditions. Cultivation, you will come to learn, is not control, it's cooperation. Our role is to create supportive conditions. And so, yes, we are collaborating with our mushrooms' growth. We are creating the scene for them to thrive through sterile practices, cleanliness, warmth, light, nutrients, all of these things to create supportive conditions for the fruiting bodies to emerge. And so safety and ethics, I think, are so important when cultivating and wild foraging. So I'm just going to jump into it here. First and foremost, safety. You need to be sure about proper identification. It's essential. Never consume unknown mushrooms. When in doubt, throw it out, or don't even pick it if you're unsure. Sometimes you do have to harvest to look at the gills, which can differentiate between certain varietals of mushrooms, or also taking a spore print, but we are not consuming anything that we are not absolutely certain of what it is. We're going to learn from trusted sources and experienced guides, practice sterile technique in cultivation, respect personal health considerations and allergies. Ethics are essential in this practice. We harvest sustainably, take only what you need, leave ecosystems undisturbed, respect private property and local regulations, honor indigenous and traditional knowledge, share wisdom responsibly, and then community responsibility. So knowledge carries influence. Teach and practice with care, protect people, ecosystems, and the integrity of the field. And that's really the role I am stepping into here as an educator, to share this information with my community, empowering you to have the confidence to grow your own medicine or food, and to properly identify different mushroom varietals in the wild. So just to review the mycelial mindset, growth happens out of sight. Connection sustains life. Transformation is constant. And studying fungi teaches resilience, cooperation, patience, and interdependence. And so this picture here on the top, you can see those little white mushrooms forming. Those are known as pins, the first little fruiting bodies to pop up, pins. but underneath is the mycelium, and it's that thread-like structure that is doing so much more communication than we're aware of on the surface. Um, and then the photo below that is a picture of space. And I'm always struck by this because not only does space look like mycelium, it also looks like brain synapses. And so we are also part of this interconnected web with the way mushrooms are now being shown to work with our neuroplasticity and consciousness, and how it's all connected to this grand design that we are part of. It's truly miraculous when you stop and think about it. Um, okay, so that was pretty much the 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 chunk of the lesson that was more structured. And here I'm just going to chat with you a little bit because I I know that this workshop is a lot. I've been uploading all the lessons to our classroom, and there's a lot of information, but I'm here to be your mentor, to coach you along. Um, and I just want to talk about gear for a second. So, with mushroom cultivation, there are two roads we can go here. So, I think it's really important to decide if you are going to sterilize or not. So, what I mean by that is sterilizing with a pressure canner is a really empowering skill. Once you have that down, you can do so much with it. You can preserve food. You'll be, in our workshop together, you'll be sterilizing grains or agar for your mycelium to live healthily without contaminants. But it can be a little overwhelming, or just, you know, just kind of scary to dive into a new tool like this. So I just want to say that if you're a little scared, that's okay. Um, I'm here to teach you about proper use of a pressure canner, and you also don't have to use it if you don't want to. Nowadays, this is really a newer thing. You can find companies that offer pre-sterilized agar and pre-sterilized grain bags with substrate. So you can still do a lot of this mycology, technical work, but you receive things that are already sterilized so that you can skip that part. And yeah, I just wanted to say that. So if you are held up by the, learning the art of pressure canning, sterilization, it's okay. You can get those items, the agar, the grain bags, and you can inoculate them yourself, or you can get them already inoculated. So like I showed in my last presentation, the masterclass, the Lumina mushroom all-in-one grow lab, that has been sterilized and inoculated for you. All you have to do is introduce the fruiting conditions, which is so easy. You just have to keep it warm, keep it in ambient light, and talk to it, communicate with it, have it in your everyday life, just so you're observing it. You can put it on your altar. But that's like the easier route. It's not any less special than starting your cultivation practice from, let's say, a spore print, or, starting out working with the mycelium that you sterilize the agar for. I'm showing you all the techniques to help you successfully pick mushrooms one day and, forage them later. So you decide Are you going to learn the art of sterilization, or do you want to buy pre-sterilized items? In our classroom, I offered some resources. So our friends at Twin Flames Mycology offer pre-sterilized agar grain bags and our Lumina Mushroom Lab, but also a website called Spore Swaps. I linked that in our classroom as well. It has so many resources on there, and it's different vendors, and so I'll also link my favorite vendors who I've worked with, but you can buy what I'm going to get into next. So let me go ahead and transport us to the next slide here. So with spore swaps, you can get the photo here, spore swabs, which are spores just I don't know, I'm having a brain moment, but okay, so you have a Q-tip and you put it in a spore print, and then you can transfer that to agar. It's just an easier way than taking the spore print, for instance, and scraping that with a tool. They come already on a tool, and then you would transport that to agar, or you can dip it into your substrate, your grains. So we're getting into all of this as we go, especially during Q&A, but I'm just sharing the different things that you will find on these websites that you're going to want to eventually obtain. So in the center here is an active culture of Mycelium on agar. This is my favorite way to work. You can order them already like this, like the picture, like on agar with the mycelium already growing, and it's a great way to get new genetics and to see that they're clean. If there was a contamination on that, it would be really noticeable. Whereas in your spore print or your spore swab, even your liquid culture, having a contaminant, you wouldn't know it until much later because it takes time for the contaminant to grow. Basically, your mycelium succumbs to a contaminant. Otherwise, your healthy culture, your mycelium grows and grows. And so I really just like using those mycelium cultures on the agar because if there's trichoderma, which is a type of mold, you see it and you know that you don't want to use it. This will save so much time in the long run. But all of these things are wonderful ways to get genetics and, to test your liquid culture or your spore swab or your spore print. You can put it on agar yourself. In 2 weeks to a month, you will have growth that you can tell if it's clean. And I know I'm using a lot of new terminology, but this will all become second nature to you as we go. Well, that is the end of my slides. And so I'm thinking here if there's anything else I'd like to say, but no, I think that's it for our first module. I encourage you to look at all of the resources that I've added to our classroom, and next week we will have our first Q&A session. I'm going to come with a short grounding meditation and also some journal prompts that we can talk about, or you can come with your questions. But it's just a sweet space for us to connect and build our community and share knowledge. And so I really hope to see you there next week, Thursday, 6 p.m. Also, the first Sunday of the month is just for the ladies. Girls Who Grow, women's group. The first Sunday of every month is just for the women. I will also be updating the calendar in our classroom as well, so you will have all of this laid out for you. I'll get that all set by the weekend. So, all right. Thank you so much for being here. I appreciate your presence. Thank you.